In modern professional and academic environments, the keyboard has almost entirely replaced the pen. We communicate, learn, and process information through our screens, driven by the need for data transfer speed. Yet this physical efficiency creates a paradox. While typing is faster, electrophysiological data reveals that it engages a limited range of neural network connectivity compared to manual writing. When we examine the biomechanics of cursive handwriting, a different picture emerges. The intricate, continuous movements required to form letters by hand involve widespread connectivity across the brain. Specifically, high-density EEG recordings show dense coherence in the theta and alpha frequency bands, concentrated across the brain's central and parietal regions. This low-frequency synchronization operates as a prerequisite for memory formation, serving as a signal that the brain is ready to encode new information. Beyond memory encoding, the activation patterns induced by handwriting are linked to the processing of complex emotions and long-term neuroplasticity. The keyboard, by contrast, requires a uniform press of a single finger. It does not activate the same neural pathways seen during manual letter formation. By prioritizing the keyboard, we choose raw operational speed over cognitive integration and emotional regulation. To observe the neurological differences between these two inputs, researchers monitored 36 university students utilizing a 256-channel high-density EEG sensor net. The participants completed trials under two distinct conditions: handwriting words in cursive using a digital pen directly on a touchscreen versus typing those same words on a standard keyboard. This connectivity matrix visualizes the contrast in brain activation. For handwriting, dense nodes of activity appear in the parietal and central regions. For typing, it remains sparse. Analyzing this reveals 32 significant cluster differences exclusively in the handwriting data. This synchronization occurs across the parietal and central regions, areas associated with visual perception, attention, and language. The activity is recorded within specific frequency parameters, beginning with the alpha band, spanning 8 to 12.5 Hz. Brain connectivity in the alpha band correlates with task-specific long-term memory performance. The second parameter is the lower frequency theta band, which operates between 3.5 and 7.5 Hz. Theta connectivity supports the brain's ability to apprehend and integrate novel information within our working memory. These EEG matrices indicate that typing fails to trigger the low-frequency networks required for the brain to integrate and retain new information. The disparity originates in biomechanics. A keystroke is binary and repetitive. Cursive writing demands complex physical integration. Cursive handwriting is a continuous spatio-temporal task. To form a single letter, the hand executes a specific, intricate pathway of motion. This forces simultaneous coordination, processing visual input while executing motor commands and evaluating proprioceptive feedback. This friction-heavy motor demand is the trigger that generates slow theta connections across long distances in the brain. In neuroscience, the presence of these slow waves is necessary for a process called a gating mechanism. Because of their low frequency, theta waves act as a communication bridge, serving as a carrier wave that links disparate regions of the cortex. By synchronizing the broader network, these slow theta waves gate or permit the occurrence of much faster gamma oscillations, which operate above 30 Hz. These high-frequency gamma oscillations are the signals responsible for episodic memory formation. Keyboards bypass the complex spatio-temporal friction needed to spark this chain reaction. Without the kinematic demands of handwriting, the brain lacks the foundational theta waves required to successfully encode long-term episodic memories. This physical act of writing is equally significant in the processing of emotional states. Journaling forces the writer to take internal, abstract emotional experiences and translate them into physical, structured text on a page. Neurologically, the process of putting feelings into exact words is known as affect labeling. Identifying and naming an emotion directly engages the right ventrolateral prefrontal cortex, or RVLPFC. As you write, it processes the structural narrative, organizing chaotic feelings into coherent linguistic patterns. Once engaged, the prefrontal cortex transmits inhibitory signals downward, targeting the amygdala, the brain's primary threat detection center. This mechanism modulates the physiological stress response within the body, lowering heart rate and reducing anxiety. Therapeutic journaling enables the executive control center to modulate the brain's baseline alarm system through the demand of emotional translation. To understand why this creates permanent change, we must look at the hippocampus, which stores and reviews past emotional events. Memories are not static. They are subject to a biological process called memory reconsolidation. When you write about a past emotional event, you pull that memory from static storage, making the neural connection temporarily malleable. While the memory is in this state, the prefrontal cortex can update the original experience with new cognitive context and rational organization. This physical updating process prevents the brain from treating looping thoughts as unresolved problems, which helps break repetitive loops of rumination. Sustained journaling restructures how the adult brain stores trauma and stress by facilitating this active reconsolidation process. Journaling and handwriting should be viewed as active structural interventions rather than passive recording tasks. The physical motor demand of the pen generates the theta and alpha synchronization required to connect disparate regions of the cortex. This synchronization primes the hippocampus for memory reconsolidation. Simultaneously, affect labeling utilizes the prefrontal cortex to modulate the amygdala, reducing the biological stress response. The result is an aligned default mode network, shifting the brain from anxiety into productive self-reflection. Typing on a keyboard is optimized for data transfer speed, but it bypasses these required neural pathways. For cognitive integration, memory retention, and emotional regulation, the physical friction of the pen remains a neurobiological necessity.